Hello, welcome to the Sound of Science AP edition. Today we're going to be talking about cells, at least an introduction to cells. We're going to talk about the origins, we're going to talk about organelles, and we'll talk a little bit about organization and how all those organelles work together. To begin, cells are believed to have all come from a common ancestor. And we believe this because all cells have both a genome, some type of DNA, and they all have ribosomes. Every cell has ribosomes. So that's why we think that cells probably share a common ancestor that had both components. There are two main types of cells that we talk about: prokaryotes, which don't have any membrane bound organelles, bacteria, for instance, and eukaryotes that have several membrane bound organelles. And we're really going to focus on eukaryotes with this. If you take a look at the prokaryote that I have here for you, it essentially has a cell wall. It has the plasma membrane around it, but once inside, you basically have the genome and ribosomes. There's not a whole lot else going on inside the bacterial cell. Makes them very, very easy to replicate. They reproduce very quickly, sometimes as quickly as every 20 minutes. And really, despite the amazing things that bacteria can do for us and can do for life on the planet, they're really pretty simplistic. Eukaryotes, on the other hand, several membrane bound organelles, much more complicated, much more complex, and much more highly organized, which is necessary for all those organelles to communicate with each other and to work together. So let's talk about that first part that that cells all have in common, these ribosomes. No membrane to these ribosomes. They actually have two subunits that are not membrane enclosed. And those two subunits are made up of proteins and something called ribosomal RNA, which is rRNA. Ribosome's whole job is to make proteins according to the cell's genome. So the genome gives the recipe or the directions to the ribosome through something called mRNA. It's made as a copy of DNA. If you remember back to protein synthesis, back from your biology class, and that should be somewhat familiar. But regardless, it creates this long chain of amino acids, a polypeptide chain, and that long chain of amino acids is ultimately going to be folded and modified and become a protein. And proteins make everything happen. All of the traits, all the characteristics, all the things that cells do are directed by proteins. As we move into the other organelles, I am going to point out an incredibly important statement. Structural difference equals functional difference. The structural difference in organelles or between organelles or even the same organelle having different structural components indicates its function. And so that's actually a really important component of what we're going to be doing today, is talking about how the structural differences lends itself to doing different jobs within the cell. So endoplasmic reticulum is one of those that I like to talk about because it actually has two different structures. We have a smooth ER and we have a rough ER. And the smooth ER is really more about mechanical support and cellular transport. That's its job. It moves things throughout the cell. The rough ER, on the other hand, is typically going to surround the nucleus, which is where the genome is stored in eukaryotic cells, and is dotted with ribosomes. That's actually what gives it its rough appearance. And so those ribosomes are there. Well, if you remember back to the previous slide, ribosomes, their job is to make proteins. So the rough ER actually aids in protein transport throughout the cell. The next organelle I want to talk about is the Golgi apparatus, or you might have heard it called the Golgi body. And the Golgi apparatus is basically just flattened series of membranes. Its whole job is to correctly fold those amino acid chains and chemically modify those newly synthesized proteins to make them the proteins they're supposed to be. And then on top of that, it's responsible for some of the protein transport, including using secretory vesicles to remove the proteins from the cell itself and actually secrete them into the surrounding areas. Lysosomes are one of those organelles that students don't typically think about as often. Lysosomes are actually going to be created by the Golgi apparatus, and they're filled with enzymes. Their job is to use those hydrolytic enzymes to do hydrolysis and break down substances. And they might be breaking down a virus that's entered, or they might be breaking down a bacteria that's infected a cell, or they may be digesting damaged cell parts. Or they might break down macromolecules such as carbohydrates or lipids and send those components that are necessary, like glucose, to the mitochondria for cellular respiration. So lysosomes are versatile. They are found in plant and animal cells. What we talk about with lysosomes is we always sort of think of them as like the garbage collectors, but really they are doing more than getting rid of waste and breaking down old things. They're actually a key component to the metabolism and metabolism activities that are occurring within the cells. Vacuoles are another one of those really versatile organelles. They can store waste until you get rid of it. They store water, and they can actually store macromolecules, like such as food. So we often think of vacuoles as storing food, water, and waste. Usually not all together. In animal cells, vacuoles tend to be numerous and small. Lots of vacuoles, tiny vacuoles, and they store individual components. In the plant cell, it's large. Usually, a large central vacuole is found in the plant cell, and it has a job that is more than just storage of water, waste, and macromolecules. It's actually responsible for keeping the plant turgid. So, turgor pressure is the force that a full vacuole inside of a cell exerts on the substances inside that plant cell, pushing out against the cell wall. This helps for plants to maintain their shape and helps keep them upright. So if you look at the diagram I've shown you here, and I do have several diagrams here, but the one to the left where it talks about high turgor pressure versus low turgor pressure, you actually can see the vacuole inside of the cell where it's full of water. The plant is standing upright, it looks good. Where it's low on water, the plant is wilted. This is why plants wilt when they don't receive enough water. If you look at the picture that's right beside me, that's actually a microscope picture of a vacuole. And that it's fairly full, but you can see how that membrane is sort of pulling away. If it were to shrink, then the actual cell itself would start to cave in, almost like a thin cardboard box can be easily pushed in. Now we've come to the part of the cell that we normally think of as being the most complicated. And the first is the mitochondria. Everybody remembers the mighty mitochondria, the powerhouse of the cell, which you will remember until your dying day, that the mighty mitochondria is the powerhouse of the cell. But there's more to it than that. And actually, mitochondria create ATP through a process known as cellular respiration. It uses the energy from macromolecules. It has a double membrane. The outer membrane is smooth. The inner membrane is folded. This is called cristae. That inner membrane being folded increases the surface area, and it allows for more production of ATP. And as a reminder, if you've forgotten, ATP just means adenosine triphosphate. This is the molecule that we use for energy in cellular activities. The Krebs cycle or citric acid cycle occurs in the intersection there, called the matrix. The electron transport chain and ATP synthesis is going to occur also in that inner membrane, but not in the matrix. So again, that increased surface area due to all the folding is really beneficial and allows us to create more ATP. which is a good thing. It's important. Without enough energy, we end up in sort of a metabolic distress. The last organelle I'm going to talk about today is the chloroplast. Now, the chloroplast is not found in all cells, and not even all eukaryotic cells. It's only going to be found in cells that can do photosynthesis, so plant cells and some protists. It also has a double membrane, just like your mitochondria. And actually, we're going to find that the chloroplast and the mitochondria are really tied together, despite the fact that sometimes they're not even in the same cell. The smooth outer membrane is there, and then you have your stacks of membranes, these little discs that are inside. Each of those is called a thylakoid. And once you get a whole stack of it, they're called grana or a granum if you're talking singular. The membranes themselves actually have pigments in them. Chlorophyll is one of those that we talk about. And chlorophyll allows for the photosystems, that's portions of photosynthesis, to take place. Chlorophyll happens to reflect green light. That's why plants appear green. Chlorophyll appears green. An electron transport chain, which I always put ETC, can be found between the two photosystems. It's embedded in the thylakoid membrane, and this is also where our light-dependent reactions are going to take place. In a later video, I'm going to talk about photosynthesis. I'm going to talk about cellular respiration. I'm going to get into more of the metabolism processes that cells experience. But for right now, I just want you to understand the parts of the cell. Our final topic is the interaction that occurs between organelles. And I told you I would talk about organization, so here it is. These interactions that occur between the organelles are vital to each of them doing their job. One of the things that I want you to understand as you go through is that just recognizing an organelle in a diagram is not enough. You have to know the function of the organelle in order to do well in AP. And so making sure that you know what each of them does is really important. When we talk about the interactions between them, it's based again on their function. And I already hinted at this a little bit when I talked about how the proteins are created sort of near the endoplasmic reticulum in those ribosomes, and then they find their way to the Golgi complex where they're folded and chemically modified into the new protein that's trying to be created. And then the Golgi complex sort of scoots it where it belongs. Sometimes those proteins are building other organelles. Sometimes they're used intercellularly. And so that's actually important to understand as well, that they might end up in other places. So I also have an animation. So I'm going to give you a chance to watch that animation. And hopefully this will sort of explain what I'm talking about.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
We've come to the end of our time. Just in summary, remember that all cells have a common ancestor. So all cells now have ribosomes and a genome, just like their common ancestor did. Eukaryotic cells have membrane-bound organelles. Prokaryotic cells do not. Organelles interact with each other within the cell. And plant and animal cells have several differences, including cell walls, chloroplast, and vacuole size. I hope all of this has made sense. If you need to go back and watch any portions again, please feel free to do so. Take great notes. And as always, I'll see you next time on The Sound of Science.